The ISB Cancer Genomics Cloud is designed to support a variety of users, from the bench biologist to the computational scientist to algorithm developers. This is accomplished through the combination of custom-developed tools such as the ISB web app and APIs with existing Google Cloud tools such as BigQuery and Compute Engine. This approach of using existing open standards and tools and developing only when necessary has created an analysis environment that is easily adapted to any workflow. The ISB CGC web app is a graphical user interface that allows users to create patient cohorts based on a variety of clinical and experimental factors and then analyze and visualize the results. The web app is comprised of several different sections. The Save Cohort section shows all of the patient cohorts you've created, as well as allowing for the creation of new cohorts. The genes and miRNA favorites, as well as the variable favorites, allows users to save gene lists, miRNA lists, and lists of important clinical traits that they can then use later in their analyses. And finally, the Saved Workbook section is where cohorts and favorites lists are combined to allow analysis and visualization. The web app revolves primarily around cohorts, and those can be created by clicking on the Create Cohort link. Once in the cohort page, simply select the program you wish to create your cohort from, be it TCGA, CCLE, Target, or your own user data. And then you can use various criteria such as case-based information like vital status, gender, age at diagnosis, or data availability. Click on the Data tab and you can see all of the data available for that program. If you wish to look at specific mutations or create a cohort from specific mutations, you can click on the Molecular tab. Enter in the gene name that you're interested in, or gene names. Here we're just going to use TP53 as an example. And then select the kind of mutation that you wish. You can use any, non-silent, or if you wish to tailor the list, you can click on Individual Selection. That allows access to a variety of different kinds of mutations that are available for that particular program. In this case, we're simply going to select the missense mutations from TCGA. Once the system has returned the data, you can save this as a new cohort by clicking on Save as New Cohort, give the cohort a name, and then create the cohort. This will save it as a cohort within your system and allow you to analyze it later on. After you've created your cohorts, as well as your genes and variable favorites, you can use the workbooks to analyze and visualize these. Workbooks work much like a spreadsheet does in that you can have multiple tabs, each holding their own data analysis. The various visualizations that you can use within the ISB CGC can be seen by clicking on the menu, then Analyses, then Browse All Analyses. This shows you that we support bar charts, histograms, scatter plots, violin plots, cubbyhole plots, and seq peaks, which show you where mutations lie along a gene. Once you have your workbook set up, simply select the kind of plot you wish from the drop-down menu. Then use the links to populate your genes and miRNAs, your variables, and your cohorts. Once you've done this, the system will ask you for which variables you wish to have plotted and then present you with the results. If on this graph you see some cases that may be interesting, you can create a cohort directly from the graph by clicking on Enable Sample Selection and then dragging around the area that contains patients that may be interesting to you. Click on Save Cohorts, then give it a name that's useful to you, and click on Save Cohorts. Cohorts saved in this manner will behave exactly like every other cohort within the system. Finally, the Save Program section allows you to bring in your own data and compare that to target TCGA and CCLE data. This data, once you've incorporated it into the system, will behave exactly like the data that ISB CGC provides and remain unique and private to you until you decide to share it with your colleagues. Google's BigQuery database allows users to analyze data using standard SQL queries without having to go through the trouble of downloading data and setting up their own database. The BigQuery interface is accessible via a web browser from the Google console, and ISB has populated BigQuery with a variety of data that are ready to use, including data from TCGA, Target, and CCLE. In addition, we've included data from community standards such as GenCode, Ensemble, Go, and Merbase, and recently have added data from the Cosmic Mutation database and made that accessible to registered Cosmic users. BigQuery also allows users to create private tables containing their own research data, making analysis of public and private data very straightforward. This mix of data allows users to perform both single cancer and cross-cancer queries with equal ease. As an example of how to use SQL within BigQuery, we're going to go through an example that shows how to combine somatic mutation data with gene expression data. In standard SQL, we can create a series of subtables and then use the data stored within those subtables to perform a final aggregate analysis. So to start, we use the with keyword followed by our first table, mutated samples. This table will contain a list of sample barcodes that have a somatic missense mutation in the EGFR gene. We are being tissue and disease agnostic in this query, so any tissue from any of the TCGA cancers that have a mutation in the EGFR gene will be included. In the second table, we're going to take the barcodes from the first table and collect the gene expression data associated with those samples. Now, BigQuery contains a number of built-in functions, and in this table, we transform the expression values with the log 10. In the final table, we make a change to the where portion of the query and get the gene expression data for the samples that do not have an EGFR mutation. This clause also highlights the built-in string handling functions, and we use regex to make sure that only the primary tumors are included by checking for the presence of the 01A string in the barcode. In the final section of this query, we compute aggregate statistics over the subtables that we've just created. We have expression from samples both with and without EGFR mutations, and we perform a union that joins the two subtables row by row. We also use additional built-in functions to perform the aggregate calculations, such as standard deviation and average. When this query is run, it will process a little over 7 gigabytes of data in just a few seconds and produce the results shown here. So while this query allows us to compare gene expression between two different cohorts based on a mutation pattern of a single gene, you can easily see expanding this or altering this to do a similar analysis on other data types, such as methylation levels, miRNA expression, or protein expression. Using the provided data and built-in functions allows users to do very sophisticated analyses without having to go through a great deal of trouble and preparation. Researchers can spend more of their time developing their queries, not organizing their data. We have a significant number of examples in our help documentation and have a Query of the Month Club, which highlights using BigQuery to analyze cancer data. The ISB Cancer Genomics Cloud includes a series of application programming interfaces encompassing many of the functions used in the web app and making them available to end users writing their own programs. When combined with the existing Google APIs, end users can take advantage of the full breadth of ISB CGC from within their own programs. In addition, the APIs and web app share cohorts, so a cohort created using one tool can be seen and used in the other. The easiest way to become acquainted with the APIs is through the use of the Google API Explorer, a web-based tool that allows you to construct queries, including filters to tailor the data being returned, and see both the query URL and the results. The ISB CGC API is divided into 4 endpoints, one for each of the programs, TCGA, Target, and CCLE, and one common endpoint for cross-program functions such as listing, getting, and deleting cohorts. The program-specific APIs allow users exploring cases and samples from that program to see what additional data is available about them, as well as to preview and create cohorts. For example, the samples get endpoint will allow you to explore the information available at the ISB CGC about a specific sample. As we mentioned earlier, you can see both the URL used to create the query as well as the data that the query returns. Using the Fields editor of the Google APIs allows you to tailor the returned data to be exactly what you wish. In this example, we'll ask for the aliquots, the case, and then some data fields such as the GDC identifier and the Google bucket location. If we rerun this query, we again see the URL used to retrieve the information as well as the reduced amount of data returned in response to that query. For the TCGA endpoints, we have included the annotation endpoints, which allow users to understand if there are any difficulties known about specific cases, samples, or aliquots. Because the APIs are implemented as REST, they can be invoked from simple tools such as curl and wget, or using any language that can implement a REST query and consume JSON content. Google provides a Python and Java discovery packages that can aid in writing programs using those languages, and ISB maintains a GitHub repository containing a variety of Python examples that demonstrate how to use these endpoints. Additional help can be found in the ISB CGC online documentation.